All right, let's talk about one of the biggest, most complex puzzles in modern medicine, schizophrenia. Now, we've known for ages that it runs in families, right? But how do you actually go from knowing that to finding the specific genes responsible? How are scientists actually cracking that code? Well, that's what we're going to get into.
Yeah, that really is the big question, isn't it? Think about it like this. The human genome is this massive, sprawling city. And the scientists, well, they're like detectives. They know something is going wrong somewhere inside the city walls, but they can't just go and round everyone up. They need clues, they need suspects, and they need a way to prove their case. So, where do you even begin? Well, their first step is to identify what are called candidate genes. And the slide says it perfectly. Think of these as the initial persons of interest in this huge genetic investigation. These are the genes that kind of get flagged, put on a watch list for a much closer look. And that brings us right to one of the first major breakthroughs in this whole detective story. And it all centers on a prime suspect, a gene called DISC1. What's so incredible about this is where the clue came from. It wasn't from some high-tech, futuristic lab. Nope. The breakthrough came from studying a single, large family in Scotland, where sadly, major mental illness was just unusually common. This family became so important to the research that they're famously known as the Scottish T111 family. And by studying this one family, researchers were able to pinpoint a specific gene. They named it DISC1, which stands for Disrupted in Schizophrenia 1. And look, this was a total game changer. It wasn't just, oh, this might be related. No, they identified it as a causal risk factor. That made it one of the single most important suspects ever found for psychiatric disorders. So why all the fuss about DISC1? Well, it's because the protein this gene makes is absolutely fundamental to how our brains work. It's a key player in building the brain's architecture and in managing all the complex signaling between neurons. So you can see why it's such a critical target, right? If you want to know how things might go wrong, you got to look at the building blocks. But of course, with something as complex as schizophrenia, it's never going to be just one thing. There's never just one suspect. So the investigation had to get bigger, and it quickly zeroed in on another major person of interest. And pretty soon, another leading candidate gene popped up on the radar, neuroglugelin-1, or NRG1 for short. Just like DISC1, this gene is super involved in brain development and function. So now, our genetic detectives had two prime suspects in the lineup. Okay, so you've got your suspects. Now what? How do you actually figure out what they do? Obviously, you can't go experimenting on human brains. So instead, scientists do the next best thing. They build a really sophisticated biological simulation using animal models. And the process is really methodical, but it's also incredibly powerful. First, they take a candidate gene, like NRG1. Then, using some pretty amazing genetic tools, they create a line of mice that have a mutation in that one specific gene. And then, they just watch. They observe them, they test them, and they compare them to their normal littermates to see exactly what's different. And when I say they observe them, I mean they measure everything. They look at the actual physical structure of the brain. We're talking about the density of connections between neurons. They measure brain function, like its electrical activity. And of course, they look at behavior, social stuff, attention, you name it. So, what did they find? What have all these models actually shown us? This is the really cool part. This is where the abstract genetic code becomes real, tangible biology. And the results have been pretty profound. Just take a look at this. It shows one of the most striking findings perfectly. There's a test that basically measures your brain's ability to filter out stuff that doesn't matter. Normal mice, they quickly learn to ignore a sound that's irrelevant. But the mice with that single NRG1 mutation, they can't. Their brains can't filter out the noise. And that directly mirrors the kind of attention deficits we so often see in humans with schizophrenia. And it doesn't stop with attention. The changes were everywhere. In the brain's actual structure, they found fewer dendritic spines. Those are the little connection points between neurons. And behaviorally, the mice were often hyperactive and had trouble with social interaction. Again, all of these things echo symptoms associated with the human disorder. Okay, so let's zoom out for a second. We've gone all the way from a family in Scotland to a mouse model in a lab. So what's the big picture here? Why is all of this so critically important? Here's the key takeaway. This research, it's the essential bridge. It's what takes us from an abstract clue, a single gene, to a real, concrete understanding of what's happening biologically. For the first time, it lets us actually see how one tiny change in our DNA can create these massive ripple effects on the brain's structure, its function, and ultimately our behavior. And that leads us to the ultimate goal. Because if we can understand the specific pathways disrupted by these genes, we can move away from guesswork and towards precision medicine. These models don't just help us understand the origins of the disorder, they become the testing ground for developing targeted, knowledge-based treatments that could one day change millions of lives. This is how the long, patient work of scientific detective work paves the way for a better future.